So this is lesson number 4 in what I'm anticipating being a 6-part series as I explore, examine, and analyze the case for the beginning or the beginnings, the debate that rages on over the biblical account of creation and what it actually says and what it doesn't say. Obviously, the secular or atheistic explanation for beginnings, how this all came to be. And obviously, whether there's a conflict really between science and faith or science and religion, or whether the science actually supports the position taken by the faithful. Today, and in this lesson, I want to look at the origin of life and the explanations that are out there and what the science and the data actually shows. I'm going to be showing a couple different videos, doing obviously some summary and explanation of my own as well. But hopefully, after this lesson is done, you'll have a kind of maybe a better understanding than you did before about, if nothing else, the complexities that are involved in life that were not well known back in the day when Charles Darwin and his contemporaries were wrestling with these topics. So again, we're going to look at the wonder of creation, and I've got 2 successive videos to share with you about this, the topic of the origin of life.
Prior to the very beginning, the beginning of our universe, time, space, and matter did not exist. Nothing. Then, Genesis 1:1 tells us, "In the beginning, God created the heavens and the earth." Since the dawn of creation, God's attributes have been seen and known by all. As a building requires a builder, creation demands a creator. An outstanding design points to a perfect designer. Our amazing universe reveals the glory of an awesome creator. God's wonders surround us, and these wonders reveal much about him. Through creation, we get a glimpse of his power and his wisdom, his majesty and his care. Creation is speaking to those who will listen. God's incredible power is displayed everywhere, from the new growth of plants in the spring to new life forming in the womb. Wherever we turn, the Creator's energizing power is unmistakable. In this episode, we will shift our attention to the creation of life. The evidence we'll be looking at is by way of molecular biology, how the densest information storage mechanism known in the universe is found in our very DNA. We will explore how simple cells are infinitely complex in actuality, and most profoundly of all, how the probability of the formation of our cells in view of the natural process model is so infinitesimally small that the mind just cannot grasp it. As we will discover, matter independently does not, nor can it, give rise to information. Much less could it create an operating system that stores this information, reads it, and sends instructions to build and operate complex DNA strands of genetic code. From one single cell, much smaller than the tip of a pin, to your entire body with over 100 trillion various type cells which are themselves encoded with the same instruction manual. It's time to take a ride deep into the cellular level. Were we just incredibly lucky? Or was there more involved than blind chance and random interactions between matter and energy? Why is chance such an unsatisfying theory? Perhaps that many analysts have done the math and understand the formidable odds against success. Chance is defined as the unknown and unpredictable element in any event that has no assignable cause or guidance from a natural law or intelligent agent. Since the 17th century, one tool has been used consistently to predict the outcome of such events. It is called mathematical probability. In very broad strokes, here's how the theory works. When we flip a coin and call heads, we know that the probability is 50% or 1 chance out of 2 to be successful. Flip the coin twice and get 2 heads in a row gives us 1 chance out of 4. 3 flips with 3 heads on a single attempt gives us 1 out of 8. Every time an additional flip is added to continue getting heads, the chances are reduced exponentially by a multiple of 2. By the time we reach 10 flips in a row, our probability is reduced to 1 out of 1024. We get the idea now. When applied to the origin of life on Earth and the random formation of large biomolecules, the probability theory clarifies that the limitations of chance is the creative agent on the primordial Earth. For example, what are the odds of a single protein could come exclusively through the blind interactions of chemistry? Our target is a smaller than average molecule made from 150 amino acids, each aligned to ensure a folded chain. Researchers have calculated the probability of success is 1 chance out of 10 to the 164th power. That's 1 correctly sequenced protein chain in 100 trillion, 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 and add another 7 trillions, failed attempts. That's 1 with 164 zeros. But despite these enormous odds, some theorists argue that, given enough time, anything is possible. So let's test the validity of this argument. First, we assume an ideal world with all the perfect conditions to provide every opportunity to succeed. We stock the oceans to capacity with amino acids. That means all the atoms on Earth, including its entire supply of carbon, nitrogen, oxygen, hydrogen, and sulfur, are present to form the 10 to the 41st complete sets of the 20 amino acids used to build proteins. Then we'll alter the laws of nature to protect these building blocks from the destructive rays of ultraviolet light and chemical contamination in this primordial soup. Now, let's turn this chemistry loose and see what happens. The amino acids start bonding furiously. An entire chain of 150 units self-assembles in only 1 second. Since all 20 types of acids are available at the majority of sites, there is a 5% or 1 out of 20 that the correct molecule will align on the chain. If the sequencing is incorrect, the chain is immediately destroyed and a new assembly begins. Throughout the planet, 6,000 million, billion, trillion, trillion attempts will take place every minute. That means in 4.6 billion years, the oldest estimated age of the Earth, the number of chains that don't fold will exceed 10 to the 58th power. It is a staggering total, but nowhere near 10 to the 164th power of trials necessary on average to build a protein with 150 amino acids by chance. Undirected chemistry cannot produce our coveted molecule during the entire history of the Earth. Scientists generally consider anything with a probability of less than 1 part in 10 to the 70th power operationally impossible. And that calculation does not take into consideration the information that's stored in the cell, which directs the cell with all aspects of operation. That estimate is solely the chance of chemicals combining to form into just something living. Now, suppose against all odds, chemical evolution produced our single protein. Would we have life? No. We'd simply only have one protein, a lifeless arrangement of amino acids. The simplest living cell that we know has over 300 different proteins. But proteins are only part of the story when we consider any actual cell. We will need carbohydrates, complex sugars, nucleic acids, DNA and RNA, lipids, and a whole variety of chemicals which jointly constitute the living state. Every one of those elements have to be brought into the same microenvironment and at the same moment in time. Each chemical building block must then be assembled and organized, the network of molecular machines that will control every facet of life. If we can appreciate just precisely how difficult it is to produce just one molecular machine using nothing except atoms and energy, we can then discover that we have a profound problem. Once we have one functioning molecular machine, you still don't have a living thing. These machines need other molecular machines to survive and replicate themselves. Even if nature was capable of producing all the molecular machines necessary, that still wouldn't be enough. They have to all be together inside this tiny little membrane bound in space that we call a cell. And it all has to come together at once. That means any of these machines were to miraculously be produced, they wouldn't survive long enough waiting for others to be produced. They would have to go on through the entire sequencing again and again until it was all created at once. This already appears mind-blowingly hopeless, yet if you feel there's still hope, then let's continue. Everything inside the cell to live needs to work together in a causal loop. The higher level of organization transcends the pieces. The spatial organization in the cell requires that molecules end up in the right place and the right time. The DNA is copied into RNA. The polymerase that does the copying has to find the right location in the DNA to start copying. The RNA has to somehow hook up with each successive sequence that is perfectly coordinated. This is an awful lot to account for by random chance. The precise sequencing for proteins to form are determined by the precise arrangement of the bases in the DNA molecule. That means that this arrangement constitutes the code for the arrangement for the amino acids in the chain. Proteins without DNA could not exist, nor could DNA exist without the proteins. They have to be in existence together at the same time, and they had to be functioning within the cell at the very beginning of life. It could not have evolved because they require one another for sustenance and utility. They could have only been created simultaneously and instantaneously. Charles Darwin himself stated in his book On the Origin of Species that such structures could not hold up if it could be demonstrated that any complex organ or structure existed which could not possibly have been formed by numerous successive slight modifications. My theory would absolutely break down. Therefore, it should be stated that the naturalistic view on random processes and evolution is unscientific. With what we have just seen in the extreme complex codes of information that could not have arisen out of pure chance, then the question is, does the complexity of DNA point to an intelligent designer? The code can only be compared to what we could see today in digital nanotechnology that directs all the proteins, sugars, amino acids, etc. in order to stay alive. Evolutionary biologist Dr. Richard Dawkins concedes that DNA is full of digital code, stating, "The machine code of the genes is uncannily computer-like." Bill Gates has said, "Human DNA is like a computer program, but far, far more advanced than any we've ever created." What are we essentially dealing with within this cell of information? To answer the question regarding the origin of life, we have to reconcile what the origin of information is. German scientist Bernd Olaf Kuppers himself stated that the problem of the origin of life is clearly basically equivalent to the problem of origin of biological information. If you want to give a computer a new function, we need to program it with a new code, provide it with new information. It is the same thing that is true in life. There's a connection between information and function. This DNA enigma is what has to be ultimately solved, and we are looking at the very nature of what information is. We can then determine that, by definition, according to the Merriam-Webster dictionary, it is communication or reception of knowledge or intelligence. Knowledge obtained by instruction. The human genome has 3 billion characters of information in DNA. As outlined in my 2020 release, Better Than a Spear in the Head, we looked at the various types of evolution and that the only type that we can agree to is microevolution because this is the only one that's been observed. This is merely just adaptation within a living species to its environment along with the various characteristics that come about in its offspring. The problem with successive offspring from one generation to the next is that it loses information rather than introducing more to it. In 1957, Francis Crick, one of the scientists that discovered DNA strands inside the cell, proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This language is what was responsible for directing protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During the process of transcription, the polymerase produces a single-stranded copy of the original instructions. This transcript then moves on into a molecular machine called a nuclear or complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. This RNA of assembly instructions is then exported out of the cell through a special gateway called the nuclear pore complex, or NPC, and into the cytoplasm. It arrives at a 2-part chemical factory, the site of a protein synthesis. This is where the process of translation begins. Once the construction of the chain is completed, it is transported into a barrel-shaped machine where it is folded into the precise shape required to perform its function. From there it is released into the cytoplasm in order to do its job. It is all an extremely well-designed system with countless moving parts that relies on each other for functionality. One would have to be quite blind not to recognize that it all requires a designer, no different than expecting an intricately built mega machine like a fully loaded Maserati Levante GTS. Take away any one part from the engine and you have yourself a useless car that wouldn't even fire up. One final piece of the puzzle that needs to be looked at here is the bacterial flagellum. Any level of research on its many moving parts, you'll discover that this biological miracle contains a rotor, stator, driveshaft, motor, inner and outer membranes, hook, and filament that is essentially a self-propelled mechanism for travel through liquid for various functions, such as in females' reproductive tracts. The flagellum has all the essential parts of a propulsion system like that of an outboard motor. When you look at how the flagellum is assembled, you can clearly see a mechanical device with a rotor, a gear wheel, and a motor that turns and whips the tail to propel itself, a far more effective design than twisted air screw blades. In a term coined irreducible complexity, this device itself would have had to be fully functional because of its multi-component parts, all of which are required to function. If you remove one part, you lose the entire function of the system. As with the example mentioned within a woman's reproductive tract, this system would fail to reproduce, hence no further generations could arise to evolve any further. In the bacterial flagellum motor, there are a total of 40 different protein parts that is necessary for this machine to work. Put simply, it is impossible for the flagellum to have evolved because, as a perfect example of irreducible complexity, It could not have functioned without any other parts. They must have fully formed and fully functioning machines, and without these flagellum, biological structures could not survive no matter what the evolutionists claim. In no peer-reviewed secular paper will we find any claim that the type 3 secretion system, or the T3SS, could emerge through functional intermediate stages. In a mutant strain that scientists devised, no relic structures were seen in their findings, meaning that a confirmation was made that these structures cannot be assembled in intermediate stages, or evolved. In Darwin's day, the cell was thought to be just a simple sac filled with jellied carbon. However, after 150 years of cell research, we've discovered that they are hugely complex. Many thousands of different operations are taking place continuously within each of the cell's main parts, called organelles. Thus, the cell cannot live until all these parts are working properly. Even the simple E. coli bacterium has 4,000 genes. If we were able to magnify the DNA of the E. coli to the thickness of a clothesline, it would be 5 miles long. Therefore, there's no such thing as a simple cell. In conclusion, our living cells could not live without all these thousands of parts. Shows that the cells were created in their finished form, just as the Bible says in Genesis 1. Even bacteria declare the glory of our Creator. Join me next time in part 2 of this Creation of Life series, where we will continue with DNA and the miracle of the cell. In part 1 of the Creation of Life, we talked about protein formation, the very first step necessary for life to form naturally, and the formidable odds against the probability of the natural process being responsible for life to arise. Our DNA is the blueprint inside every cell that virtually explains how to build all the proteins in our body. Inside is encoded the very language in which God used to create life. Inside every 100 trillion cells in your body include the DNA in which we find this incredible coded language. If we were to open up this double helix and stretch it out, it would span 6 feet in length. This is found in every single one of our cells. This code is comprised of a 4-character chemical alphabet that precisely spells out the assembly instructions for all the thousands of different proteins in how your body is constructed. In fact, DNA is the most efficient and most dense information storage system in the known universe. Modern science cannot come anywhere near in replicating what the DNA can do in containing information. Picture a teaspoon of peanut butter. Imagine that this peanut butter represents pure DNA. How much genetic information can this teaspoon of DNA hold? It can hold all of the precise assembly instructions for all of the proteins of every one of those 100 million species of animal life on Earth that have ever existed. And it would still have the capacity left over for all of the information in every book ever written in human history. This is what we call unbelievably stupendous efficiency. Not only are these DNA able to replicate themselves quickly and efficiently, but it is also programmed with mechanisms that can detect and correct replication errors. They are self-correcting. There are special proteins called enzymes that actually move up and down the DNA molecule, seeking out and making repairs on a second-by-second basis. It was magnificently designed with editorial capabilities like an editor who reads through newspaper articles correcting any mistakes before being released. In the scientific study called information science, one of the laws of information science is that information never originates spontaneously in matter. If we were able to trace the copying of information back to its source, it always comes back to a mind. So since we have a creative information in DNA, That tells us it comes from intelligence. It is not something that can possibly come through millions of years of mutations or natural selection. It simply will not work. Even the DNA molecule is simple when compared to cells. Each cell functions as a city. When considering how the human body is comprised of trillions of cells working together as one unit, we should be in humble awe of our Creator's intimate care and perfect wisdom. So let's then take a glance at what these cell cities look like. When we look within a cell, it is not just a simple blob with a few strands of DNA floating around. It is important to truly appreciate the complexity of what is going on inside. It is like a highly organized city with a transportation system and infrastructure. Workers, or proteins, work in a power plant or mitochondria. And trucks, kinesin and DNA, travel along roads, actin fibers and microtubules, whose cargo are containers or vesicles that travel from factories or ribosomes that make proteins. There is a library, a genome, post office, inside of which the Golgi apparatus sorts, packages, and modifies macromolecules for secretion or for delivery to other organelles. And we have a police, the chaperones. Wayward or malformed proteins get a ticket, ubiquitin, and then get shipped off for degradation. Cells are also involved in exporting and importing. Communications and regulation occurs through complex signaling pathways that help keep everything running smoothly. Still think this all came about by chance mutations? Inside a cell, things can be transported in two ways. The first is by diffusion, in which small molecules are released and just wander around. This is usually used for small molecules like ATP, which is the fuel that is used to power molecular motors. The second way is through active transport that works like container shipping. Container shipping revolutionized the shipping industry. Rather than stacking sacks of sugar on ships, the sacks are placed in metal containers that can be stacked on the decks of ships and then later transferred from the ship onto 18-wheeler trucks. Here's the amazing part. Similar to the shipping industry, cells pack cargo into small bags called vesicles. These vesicles have motor proteins attached to their outside. There, motor proteins have two legs and can walk just like a person, putting one foot in front of another. Can you really believe it? These walk along roads that are thin filaments of polymers. And as these walk along these roads, they haul the cargo vesicles. These cargoes can be a wide variety of things. For example, vesicles can contain pigment which, when spread out, will change the color of a skin cell. Or the cargo can be a mitochondria, which is a power plant or organelle that makes ATP. The roads are of two types. Microtubules, MT, are like national or interstate highways. They spread radially outward from the nucleus of the cell like highways leading away from a major city. Actin filaments, or AF, are like local surface streets that are randomly placed. This transportation system is also regulated. For example, pigment cells in frogs change color when they get scared by a predator. When the pigment granules are spreading out from the nucleus, they will go straight through actin filament intersections until they get to the end of the filament. When the granules need to return to the nucleus, cargoes will often switch at actin filament intersections as if to find the nearest on-ramp to a microtubule. As noted earlier, molecular motors have two legs and can take one step at a time. The motor, called kinesin, walks outwards along microtubules towards the periphery of the cell and takes 8 nanometer steps. The motor, called dynein, walks along microtubules inwards toward the nucleus. Dr. Roop Malik, at the University of California, Irvine, has found that Dynein effectively has a transmission or gear shift. It can take steps of varying sizes, 8, 16, 24, or 32 nanometer steps, depending on the load it is hauling or on the amount of ATP or fuel that is available. Under a heavy load, it takes small steps. Under no load and with lots of ATP available, it takes large steps. The book of Genesis was written by Moses somewhere between 1445 to 1405 BC. The account of the creation of Eve and Adam was formed from the dust of the ground. It was carefully recorded that God did not go back to the dust to form Eve. Rather, God chose to form Eve as a perfect and suitable helper and companion for Adam from a part of his own body. So the Lord God caused the man to fall into a deep sleep, and while he was sleeping, he took one of the man's ribs and then closed up the place with flesh. Then the Lord God made a woman from the rib he had taken out of the man, and he brought her to the man. The idea of taking a bone and growing a new person from it seems quite strange, particularly from a rib. Why the rib? Why not any other part? As we look more closely to the story, we will find out that God knew exactly what he was doing. By making a perfect match for Adam, using his DNA makes perfect sense. We see throughout creation that when God creates the diversity of animals on Earth, he doesn't go about having to reinvent the wheel, so to speak, each time one is made. Each mammal species have similar parts to them: a spine, rib cage, skull, nervous system. Thus each type is wonderfully made by him, and the DNA information in each of these creatures vary quite little within its code. By looking at the creation of Eve, God wanted to copy Adam's perfectly sequenced DNA to make her the perfectly suitable partner. Here comes the astounding answer to the question of why the rib. Only in recent times have we discovered that only the rib of the human, and with various animals that we found, is the only bone in the body that can actually grow back. Many assume that Adam lived out the rest of his life with one rib missing, but in fact, after time, it actually grew back. If the Bible was just some fairy tale fiction as many atheists claim it is, how would Moses have known this fact, being the writer? Wasn't he just some semi-illiterate goat herder inventing a work of fiction? Just more evidence here to conclude that the Bible was written under the inspiration of God, our Creator. What's more, evolution teaches that we share a common ancestor with chimpanzees. However, we now have sequenced DNA for both humans and chimps. Dr. Jeffrey Tompkins, PhD in genetics, has studied these comparisons for years and have found that evolutionists mistakenly use a very preferential and selective treatment of the data when comparing our genes. They use sequences they expect to be similar. They do not include non-aligned DNA, gaps, copy number variations, and size differences. These are legitimate and massive differences between the two which cannot be reconciled. These are massively ignored because they do not fit into the evolutionary story. So how do they make it fit? They only count one type of difference. We are told we are 98 to 99% the same as chimps. However, this 1 to 2% difference represents 450 million differences along the chain of DNA that don't match up at all. This fact is suppressed by the evolution geneticists. Studies using the mitochondrial DNA, or the DNA that was only passed down from maternal ancestry, found that dating for mitochondrial Eve calculates a date of approximately 108,000 to 300,000 years. However, this data assumes human-chimp ancestry. But in 1997, some evolutionists produced another paper stating that mitochondrial Eve was only 6,500 years old. Why the vast difference from the other findings? This paper reveals that the tests do not assume human-chimp ancestry, but rather only human. This age of 6,500 years aligns extremely well with our dating of Adam and Eve by counting back the years throughout the Bible. We have the good fortune that the Old Testament records the ages of each generation from that time, which calculates to roughly 6,000 to 6,500 years ago. We've seen how astronomically impossible it could be for one living organism to come into existence out of non-living matter, and that would be just one organism. We still haven't reconciled how millions of complex organisms could have evolved from an undirected natural process. Because natural selection does not dictate to other non-living matter in other parts of the world and across the seas on how to evolve as well. Each and every one of those events need to go through the same improbable processes as all others. Given all that we've covered above, should the popular scenario of natural processes of chemical and biological evolution be taught globally as the only explanation of the origin of life and species? It is clear that secular academia are completely unwilling to open their minds to other possibilities or other explanations. Popular science itself stresses that if observations and data contradict your testing, then the theory should be modified or abandoned. Unfortunately, however, this is clearly not happening within our present study of accepted evolution. Instead, what is happening is that these scientists take the information they receive and make it fit the evolutionary model. Is that a good scientific practice? We can see without a shadow of a doubt that there is undeniable bias going on within the scientific community. Geosociety and their definition of the nature of science and the scientific method says, to be scientific in our era is to search for solely natural explanations. What? This is an intentional exclusion based on their definition of quote-unquote true science. Therefore, before the race even begins, the winner has already been declared. Really? Defining the word science to fit your academic worldview is like defining the rules to a game to ensure that you always win. Such bias runs deep into the entire thought process about any given genetic development in living things. For example, some evolutionists have claimed that the compound I in the honeybee is a bad design that no good designer would use, so it must have evolved. However, it is actually an excellent design for small creatures, enabling bees to navigate by the highly efficient optic flow method, all centrally controlled by a navigation software packed into a brain the size of a sesame seed. Evolution scientists argue against using the term design in nature, and they go so far as to state that teaching intelligent design isn't science. However, the best robotics engineers have yet to design a navigation program as good as a bee's and run it on a chip with the energy efficiency of a bee's brain. So it's reasonable to believe that the bee was designed by a master programmer whose intelligence is beyond our own. Critical thinking is required to realize that scientific findings do not violate the principles of information science. Too often, scientists believe that information can be generated by physical processes, yet we know that simply is not possible. Functional information cannot be generated from purely physical properties. Functional information has never been observed to arise purely by physical interactions. And just to touch on one more aspect of the creation of life, we cannot fail to talk about consciousness. Life doesn't just comprise of living matter in the form of a physical being. It is also conscious. You and I are conscious of ourselves and the world around us. Now what or how on earth could give rise to that? We can see how the mechanics of protein be pulled together, something purely intangible as our sentience or awareness of existence. Most honest atheists agree that evolutionary processes cannot explain the existence of human consciousness, yet they still cling to the belief that some as yet undiscovered natural process gave rise to it. Cognitive scientist Professor Jerry Fedor states, Nobody has the slightest idea how anything material could be conscious. Not only can they not explain it, but they can't even define it. In contrast, the Bible provides a wholly satisfactory framework upon which to build and understand human nature. The scriptures tell us we are much more than just a body or a biochemical mechanism. When we were created, we were given an immaterial soul and a spiritual nature, with it fully conscious and aware mind to discern, navigate, and explore this beautiful world He put us in. Join me next time where we explore the plethora of evidence for the global catastrophe that destroyed our world.
So I hope that was helpful. It should have, at least with some of you, I know it did with me. It kind of brought me back to those uh, high school biology classes where we were learning about the components and the functions of the cell and so forth. And as he said, back when Darwin and his compatriots were coming up with their theory, they thought the cell was just basically a simple blob of tissue. But of course, with the discovery of better and better microscopes and so forth, and the ability to study the cell, obviously we've come to understand that they are extraordinarily complex. In fact, they are more complex than any computer that human beings with intelligence have ever designed. Anyway, let me just, quickly kind of um, go through some of the things that were brought up and maybe a couple different ones. But as far as the beginnings of life, as was mentioned, evolutionists adhere to a belief that life sprang from non-living matter. That I talked before in previous episode that at a point in time, there was no matter, no material universe or any matter at all. And then, of course, everything that we see, everything that we observe came into existence. Some theorize in a big bang and so forth. But basically, even then, it was just non-living matter. It was just things. And evolutionists have to, obviously, if they're going to explain things in a naturalistic standpoint, they have to adhere to a belief that somehow the non-living matter became living matter. They talk about the primordial soup, a primordial ocean that was, as was explained in part one, full of these elements, and that somehow these parts, of course, they don't explain where the parts came from, but anyway, even if they did exist, they kind of self-organized themselves and somehow became from a non-living to a living cell. However, there is really no indication, absolutely no indication of how life could have sprung from non-living matter. There is no science to support it. Obviously, they theorize it because they have to. If they're going to come up with some kind of natural explanation for everything that we are aware of, they have to come up with a naturalistic explanation. The popular conception of primitive cells as the starting point for the origin of the species is really erroneous. There is nothing functionally primitive about such cells. As we were just saying, they contained basically the same biochemical equipment as do their modern counterparts. How then did the precursor cell arise? The only unequivocal rejoinder to this question is that we do not know. And obviously, this is not a creationist talking. This is an evolutionist. They throw up their hands. We don't know. We can't answer that kind of a thing. And then DNA. The videos talked a lot about DNA. And of course, the discovery of DNA was in many respects a game changer because it is the basic information system of the cell. And it again is highly, highly complex. I'm sure you got, if you didn't know before, the descriptions and the graphics in the videos certainly pointed that out. DNA is composed of proteins, and the proteins require DNA to produce them. So, as was pointed out in the video, they had to both exist at the same time. They had to both come into being at the same time because they're interdependent. They both depend on each other for their existence. You can't have one without the other. It would require some kind of spontaneous genesis out of nothing because they are so interdependent. I got one more little video to show you. It's talking about DNA. Whoops. I guess I can't play that. Anyway, the video did point out quite a bit of information about DNA, the complexity of it, the difficulty in coming up with a theory for how it originated, and again, the fact that it had to come into being at the exact same time that the proteins it depends on and depends on it. According to evolution, life began several billion years ago, and it proceeded to advance through thousands of generations to develop from the simple forms into the more complex ones. That is basically the theory of evolution in a nutshell. And of course, the mechanism by which this is supposed to have happened is natural selection. The idea that the survival of the fittest organisms produce the most offspring. That's, again, kind of the foundation of what evolution purports. Here's a quote from Philip Johnson, who's not an evolutionist, but here's what he said in his book, Darwin on Trial. When I want to know how a fish can become a man, and of course, if you look at the tree of life, that's what they purport. I am not enlightened by being told that the organisms that leave the most offspring are the ones that leave the most offspring. I mean, that's in essence what evolution teaches. And of course, evolution depends on mutations. It purports that mutations were the means by which changes in the species occurred, little changes that accumulated over time. But there's a problem. And that is that virtually all the mutations studied in genetics laboratories, which are pretty macro because otherwise geneticists wouldn't even notice them, are deleterious to the animals possessing them. The vast, vast, overwhelming majority of mutations are harmful to the organism or the being. And therefore, for mutations to be the mechanism by which this accumulation of changes occurred is extraordinarily unlikely. One Nobel Prize winning scientist has said trying to improve an organism by mutation is like trying to improve a Swiss watch by dropping it and bending one of its wheels. Improving life by random mutation has a probability of 0. And then this one of the videos gave a quote from Charles Darwin about, the idea that if it could be proven that any organism or process could not have occurred by small micro, if you will, advancements, his theory would fall apart. Well, here's another quote from Charles Darwin. To suppose the eye with all its inimitable contrivances for adjusting the focus to different distances, for admitting amounts of light and for correction of spherical and chromatic aberration, could have been formed by natural selection seems, I must confess, absurd to the highest degree. And in fact, just as with the cell, if you look at the anatomy and physiology of the eye and all the different parts of it that have to all be working correctly all at once. The idea that the eye could have come into being little by little by little makes no sense because the eye wouldn't work at all until everything was in place. Therefore, the organism, the animal, the being, uh, would have no benefit from this advancement, these mutations, until it was all in place. And so, why would it be kept in place until it was all together? Charles Darwin, and again, this is the quote that was given in the video. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely just break down. And then Michael Denton, speaking of the bird lung, which differs from its supposed ancestors that it came down to just how such a different respiratory system could have evolved gradually from the standard vertebrate design is fantastically difficult to envisage, especially bearing in mind that the maintenance of respiratory function is absolutely vital to life of an organism to the extent that the slightest malfunction leads to death within minutes. So how a lung system could change from one species to another, the transition of that is ridiculous, really, to imagine, let alone to propose. And yet that's what they do. And of course, using that same well, we could really use that same argument, not just for the bird lung, but for many different organs and processes and so forth that supposedly transitioned, down through or up through the tree of life. So again, this has certainly not been an exhaustive study of the discussion, the debates, and so forth about the origins of life. But I certainly hope that it has, if nothing else, demonstrated the complexity that's involved in living organisms, even down to the basics of just a single cell, and that a single cell, as it said in the video, is it's like a factory or it's like a city. There's so many different processes going on, and again, to suppose that it could have occurred one little tick, one little mutation at a time is really kind of hard to believe. And yet, of course, that's what evolutionists have to propose. And one of the things that they count on is the fact that we don't know, the lay public doesn't understand how complex the single cell is, let alone looking at the tree of life and trying to grasp that all of this could have come to happen without an intelligence behind it. And of course, we know who that intelligence is, and the brilliance, obviously, that had to have been involved in this process, just creating, really, in just creating a single cell, let alone the tree of life, the complexity of life, the universe, as we know it. So I know this has been a lot of technical stuff. I hope that you found it interesting, though, and I hope that you'll look for the next video in the series. God bless you.